All right, so now we're going to talk about each individual step of cellular respiration. So we have 4 steps. They're going to be glycolysis, the preparatory reaction, the citric acid cycle, and the electron transport chain. The only one of these to occur outside of the mitochondria in the cytoplasm is glycolysis. So this step will take place in the cytoplasm. Everything else will then take place in the mitochondria. And this is seen in the figure right next to the title chapter. So the first learning objective says, what is the role of glycolysis? So this will be an incomplete breakdown of carbohydrates. We will start with an organic molecule, and we will end with an organic molecule. Whenever we talk about complete versus incomplete, so you remember cellular respiration is the complete breakdown of a carbohydrate, which means it goes from organic to inorganic. This is an incomplete because we will start with an organic molecule, glucose, and we will end with an organic molecule, pyruvate. We will also begin to create ATP. If you remember in our coupled reaction, we are trying to take the energy that is in glucose and get it converted into ATP, a usable form. We will begin to create some of those ATP molecules in this first step. For the rest of this video, the figure 7.4 is going to be very important. So we have figure 7.4. And what you'll see in figure 7.4 is we can break glucose into glycolysis into 2 steps. So the 2 steps are 1, the energy investment stage, and 2 will be the energy harvest stage. So first let's talk about energy investment. To begin this, we start off with glucose. Glucose is the reactant of the energy harvest phase. And you can see this molecule is going to be split. So during the energy investment, we will split the glucose molecule. Glucose is a 6-carbon molecule. So you notice those black dots that are in the picture. We don't really care about everything else that is in this glucose, just the carbon. So while this is glucose is C6H12O6, during these reactions, we're only going to follow the carbon molecules. We're going to use energy. We're going to use ATP to split. So ATP will get reduced to ADP plus P in order to split our glucose molecule. And when we split it, we will get 2 3-carbon molecules. So remember, black marbles in our molecule set were carbon. These molecules are called G3P. And they're called G3P because actually what ends up happening is the ATP gets reduced to ADP and the phosphates are going to get added to our G3P molecules. So G3P, these are my reactants. Sorry, these are my products. So the input of the energy investment stage is glucose. The product are 2 G3P molecules. Here's 1, here's 2. As we recall, these always go together with energy reactions. The energy from ATP is released, and that phosphate group is added here. So we've released the energy from the ATP, and we've added that energy to this G3P molecule. So now, these G3P molecules are energized. The energy that was in the ATP is now in the J3P molecule. This is why it's called an investment. We've invested 2 ATP molecules to start this off. So we get 2 ADP molecules and 1, 2 phosphates. This is just like an investment that you would have. When you go to make an investment in a bank, let's say you're going to get a CD that gets interest, you're going to have to put some money in. And then hopefully it gains interest over time and you get an energized molecule. Your money that you earn is your energized molecule. This is the first half of glycolysis. So the second part, after you make your investment, you hope to make some money on that investment. What we hope to gain now is we've put in 2 ATP molecules, so right now we're 2 ATPs in the hole. We now want to hope to get some of that energy back. And when you harvest something, it's like harvesting at the end of a season. You planted some crops, you've made an investment, now you hope to get some food back from what you had. So we're going to start with those same G3P molecules. So here's one of those G3Ps. Here is the second G3P. And remember, this is a 3-carbon molecule. This one is also a 3-carbon molecule. And they have energy from phosphorus. Now the first thing that's going to happen is we're going to not actually get some ATP out of it, but we're going to transfer some energy to an NAD molecule. So as you can see in the picture, the first thing that's going to happen is we're actually going to put another phosphate group onto each of these G3P molecules. So you notice that this first molecule only had 1 P. The second molecule is going to have 2 phosphate groups. So it turns out as G3P gets converted into another molecule, we don't need to know the name of it, we're actually going to energize another molecule of NAD. And as we do so and add that phosphate group, we're going to create a molecule called NADH. So right now, just know that this is an important molecule that we're going to come back to later. The same thing is going to happen over here. As this G3P molecule adds another phosphate group, we're actually going to lose some hydrogens from it, and we're going to create another NADH molecule. So we'll come back to NADH, but know that this is an important molecule, and it will have consequences for how much energy we can earn. Then you're going to notice we're going to take off all 4 of these phosphate groups from this carbon molecule. So we're going to transfer this molecule here that has 3 carbons, and we're going to lose the 2 phosphates over time. We're going to take this molecule that also has 2 phosphates, and we're going to lose them through lots of different reactions, and we're going to end up creating energy. Every time we take a phosphate off of each of these molecules, we will make an ATP. So 2 ADP are going to go in, and as they go in, 1 ADP will grab this one, the second will grab this one, and we will create 2 more ATP molecules. Over here, 2 ADP molecules will go in. Each will capture one of these phosphates, and we will get 2 more ATP molecules. So through this process, the G3P is going to lose its phosphate, and these final molecules are called pyruvate. So here are our 2 pyruvate molecules. So remember, we started originally with 1 glucose with 6 carbons. We ended up with 2 pyruvates with 3 carbons each. But here are the 1, 2, 3, 4, 5, 6 carbons from the original molecule. So glycolysis is all about just manipulating this molecule until we can create this energy. So overall, let's take a look at what happens for each step. So, step 1, the investment stage. We take 1 glucose, which is a 6-carbon molecule, and we convert it into 2 G3P molecules, 2 3-carbon molecules. We have 6 carbons when we start. We have 6 carbons when we end. Same thing with matter. Same thing happens with matter that happens with energy. Matter is either lost or destroyed, just change from one form to another. 6 carbons, 6 carbons. Start with one 6-carbon molecule, we change it to a different form, but we didn't lose anything. And as we've talked before, every time we have metabolic reactions happening, we also have energy reactions happening. During this part, we take those 2 ATP and they get changed into 2 ADP and 2 phosphates. So these 2 things are happening at the same time. And then during harvest, we complete glycolysis, and we start with those 2 G3P molecules, and they get converted into 2 pyruvate. And at the same time, we capture some energy. We take 4 ADP molecules and 4 phosphates, remember that we're each hanging off of these molecules, and we create 4 ATP. Now, an important distinction. If I ask you how many gross ATP are created by glycolysis, that would be 4 gross. But it's just like a paycheck. If you look at your gross pay, if you get paid $10 an hour and you worked 40 hours, you would get a gross pay of $400. But as we all know, taxes have to be taken out. Your investment has to be taken out. So if I ask you how many gross ATP are created during glycolysis, your answer is 4. But if we want to know how many net, we only get a net of 2 ATP. If we take out our investment, 4 minus 2 means we only actually made a total of 2 net. So gross versus net is going to be important. And then the other molecule that's important here is that NADH. Also, during this time, we'll take 2 NAD plus 2 hydrogen ions plus 2 electrons, and we will create 2 NADH molecules. And we will come back to those in the 4th step. So this is the totality of glycolysis. And you need to know where this step takes place. We already talked about that. That is in the cytoplasm. You need to know what the different inputs and the different outputs are and where they occur. So you can see this in figure 7.6. They give a summary of the important molecules. So inputs. We have glucose. This is in the investment portion. We invest 1 glucose molecule. Eventually, that will get turned into 2 pyruvates. This will happen in the harvest section. We will put in 2 NAD plus molecules. They will get converted to 2 NADH molecules. This is happening during the harvest. So NAD is an input. And even though they didn't put it, we also put in 2 hydrogen ions. And we also put in 2 electrons. An NAD plus a hydrogen plus an electron will get us at NADH. Because remember, H plus and electron are a hydrogen. We will have an input of 2 ATP during the investment part. And that means they will end up losing and becoming 2 ADP during the investment part. And then conversely, during the harvest station, we will put in 4 ADP and 4 phosphates, and we will get 4 ATP. And this takes place during the harvest. So those are the molecules you need to know and should be able to follow during glycolysis.